Human Genome Project is a scientific research project conducted internationally, where the main objectives and goals are being able to discover and determine the sequence of chemical base pairs which make up human DNA. The mapping and identification of every single gene of what is now regarded as the human genome is the aim for this project from a functional and physical standpoint. DNA molecules are made of two twisting paired strands. Each strand is made of four chemical units called nucleotide bases. There are four of these bases which are marked as A, G, T, and C. As you can see on this brief timeline, there was a number of vital contributions leading up to the funding of the project, including the discovery of physical maps and human genetic maps using genetic markers. This led to the funding of the Human Genome Project, with a staggering $15 billion being put towards the project in 1990, launching the start of the Human Genome Project. This led to genome databases being established in 91 and a continual development of various scientific discoveries and advancement, all contributing to the almost eventual completion of the Human Genome Project in 2003. However, research is still being conducted to the present day. The first human sequence data was published in 2001 and mapping goals were achieved in 1994, just to name some large pieces of importance in the history of the project. The Human Genome Project has a number of benefits falling into two main categories: goals for the benefits, meaning where they wanted to get scientifically, as well as the applications that these scientific discoveries led to. The main goals of the Human Genome Project consist of to identify all the genes within human DNA, determine the sequences of the 3 billion chemical base pairs that make up human DNA, store information in databases, improve tools for data analysis, transfer related technologies to the private sector, as well as address the ethical, legal, and social issues that may arise from the project. Goals provided an aim at where a project needs to get, which is exactly what these goals did, allowing these applications to occur. Extremely useful in diagnosing diseases, e.g., heritable diseases, provides genetic counseling to those considering having children, can identify human genes and proteins that can target for drugs and new gene therapies, and cloning of genes advancements. Although there are so many benefits and applications, there are also various limitations. The data that is produced only provides a starting point. It is then what we do with the data produced, as it is only a preliminary stage rather than a complete and full understanding of organism and cell functioning. We can know all the base sequences of DNA on a chromosome, but that does not determine the function of every gene, as well as the functions and biochemistry behind being human. In fact, there has been large amounts of criticisms towards the data being obtained because of these limitations, after the amount of money and funding put towards the project. This data can lead to discrimination against certain genotypes, for example, those people with a genetic predisposition to certain diseases. Guidelines addressing the legal, ethical, and social issues surrounding this project are a continual development and hot topic in the science world. One's genes cannot explain one's being. An organism is the expression of a dynamic and ongoing interaction between the state of its environment and its internal state, which includes its past history and its toolbox of DNA sequences. The genome does not function as a master plan or computer program for controlling the organism. The genome is the organism's servant, not its master. The availability of recombinant DNA technology has played a vital role, being an important factor and one of the most successful techniques in making the Human Genome Project achievable. An example of these recombinant DNA technologies includes the polymerase chain reaction, which is used to replicate numerous copies of DNA. This technique was incredibly useful as it was cheap and efficient and allowed scientists to amplify a focused segment of DNA. This technique allows scientists to diagnose a hereditary disease, as well as allowing the identification of genetic fingerprints and the diagnosis of infectious diseases. This is the process. Initialization step. This step consists of heating the reaction to a temperature of 94 to 96 degrees Celsius, which is held for 1 to 9 minutes. Denaturation step. This step is the first regular cycling event and consists of heating the reaction to 94 to 98 degrees Celsius for 20 to 30 seconds. It causes DNA melting of the DNA template by disrupting the hydrogen bonds between complementary bases, yielding single-stranded DNA molecules. Annealing step. The reaction temperature is lowered to 50 to 65 degrees Celsius for 20 to 40 seconds, allowing annealing of the primers to the single-stranded DNA template. The polymerase binds to the primer template hybrid and begins DNA formation. And lastly, the extension/elongation step. The temperature at this step depends on the DNA polymerase used. At this step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding dNTPs that are complementary to the template in 5 to 3 direction. Condensing the 5 phosphate group of the dNTPs with the 3 hydroxyl group at the end of the nascent extending DNA strand. The extension time depends both on the DNA polymerase used and on the length of the DNA fragment to amplify. Some other useful recombinant DNA technologies include DNA hybridization, southern blotting, and use of genetic probes. Genetic linkage is a tendency of alleles that are close together on a chromosome to be inherited together during the meiosis phase of sexual reproduction. Genetic linkage maps are based on the frequencies of recombination between markers during crossover of homologous chromosomes. The greater the segregation between two genetic markers, the further apart they assume to be, which correlates the same way as when the frequency of recombination is closer together between the two markers, then the distance between them physically is also smaller. Originally, the markers were phenotypes that were easily detectable, like enzyme production or eye color, which were derived from coding DNA sequences. After further advancements in technology, assumed or confirmed non-coding DNA sequences, such as microsatellites or those generating restriction fragment length polymorphisms, have been used. These markers allow scientists to roughly see where a gene lies in terms of location on a chromosome. Genetic maps are not physical maps or a gene map. However, it is very important to understand that linkage maps are unable to give the exact location of specific genes. Linkage maps also do not provide nucleotide sequencing. This results in them being insufficient in terms of completing the human genome due to the fact that they don't find the exact location of genes on a chromosome, but instead they only locate the relative position of that base on the chromosome.